listeners, welcome back to our channel Be Curious. आज हम पढ़ने जा रहे हैं सेल्स, ऑर्गन्स एंड माइक्रो एनवायरनमेंट्स ऑफ द इम्यून सिस्टम. Innate immune cells that mount a first line of defense against pathogen antigens presenting cells that communicate the infection to lymphoid cells, which coordinate the adaptive response and generate the memory cells which prevent future infections. Primary lymphoid organs, including the bone marrow and the thymus, regulate the development of immune cells from immature precursors. Secondary lymphoid organs, including the spleen, lymph nodes, and specialized sites in the gut and other mucosal tissues, coordinate the encounter of antigen with antigen specific lymphocytes and their development into effector and memory cells. All functional specialized mature blood cells like red blood cells, granulocytes, macrophages, dendritic cells, and lymphocytes arise from a single cell type, the hematopoietic stem cells. The process by which HSC differentiate into mature blood cell is called hematopoiesis. Two primary lymphoid organs are responsible for the development of stem cells into mature immune cells. The bone marrow, where HSC reside and give rise to all cell types, and the thymus, where T cells complete their maturation. Cells of the immune system. Stem cells are defined by two capacities: the ability to generate or self-renew and the ability to differentiate into all diverse cell types. Embryonic stem cells have the capacity to generate every specialized cell type in an organism. In other words, they are pluripotent. Adult stem cells, in contrast, have the capacity to give rise to the diverse cell types that specify a particular tissue. Multiple adult organs are stem cells, adult stem cells that can give rise to mature tissue-specific cells. The HSC is considered the paradigmatic adult stem cell because it can differentiate into all the type of blood cells. This picture is introduce hematopoiesis, which is self-renewing hematopoietic stem cells give rise to lymphoid and myeloid progenitor. Most immune cells mature in the bone marrow and then travel to peripheral organs via the blood. Some, including mast cell and macrophage, undergo further maturation outside the bone marrow. T cells develop to maturity in the thymus. Hematopoietic stem cells have the ability to differentiate into many types of blood cells. HSCs are there with fewer than one HSC is present per 500 into 10 power 4 cell in the bone marrow, and their numbers are strictly controlled by a balance of cell division, death, and differentiation. Other daughter cells differentiate into progenitor cells that lose their self-renewal capacity and become progressively more committed to a particular blood cell lineage. Hematopoietic system have been completely destroyed by a lethal dose of X-ray, monoclonal antibodies, and flow cytometry, and identified surface protein including CD34, SK1, and C kit that distinguish the rare hematopoietic stem cell population. Hematopoiesis is the process by which hematopoietic stem cells develop into mature blood cells. It can become a common myeloid erythroid progenitor CMP, which give rise to all red blood cells, granulocytes, monocytes, and macrophages, or it can become a common lymphoid progenitor, CLP, which give rise to B lymphocyte, T lymphocytes, and NK cells. Myeloid cells and NK cells are members of the innate immune system and are the first cells to respond to infection or other insults. Lymphocytes are members of the adaptive immune response and generate a defined antigen specific immune response that also give rise to immune memory. Interestingly, both myeloid and lymphoid lineage give rise to dendritic cells, antigen presenting cells with diverse feature and function that play an important role in initiating adaptive immune response. Regulation of lineage commitment during hematopoiesis. Each step a hematopoietic stem cell take toward commitment to a particular cellular lineage is accompanied by genetic changes.